All right, let's dive in. When you look around, you see living things, non-living things. And you've probably asked yourself, what is it that truly makes something alive? Distinct from the inanimate world. What does a living thing have that, say, a rock just doesn't?
Well, the profound answer, really, despite all the complexity, is the cell.
The cell?
Exactly. It's the basic unit of life, the fundamental building block that underlies, well, all the incredible diversity you see. But it also highlights this deep unity in every living form.
That's a really powerful idea, and that's precisely what we're going to unpack today in this deep dive. We've gathered the source materials, from the foundational theories to the nitty-gritty functions of the tiniest cell parts. We want to give you a really clear look into cell biology. Our mission, as always, is to pull out those crucial nuggets, to give you a shortcut, really, to being well-informed about something so basic yet so incredibly complex.
And it's quite a journey we'll take. We'll look at how we first came to understand cells, explore the big differences and surprising similarities between them. And then we'll peel back the layers, see how scientists use physics and chemistry, this approach called reductionist biology, to figure out the molecular mysteries. We'll even touch on a key figure, G.N. Ramachandran. His work using exactly that approach totally changed our view of proteins.
Okay, so let's start right at the beginning then. What is a cell, fundamentally? The sources are clear that even single-celled organisms can have this independent existence. They do all the essential life functions.
Yeah, that's key.
Right, because anything less than a complete cell structure, it can't live independently. So that makes the cell the fundamental structural and functional unit of all living organisms.
Absolute foundational. And the first people to actually see this world were real pioneers. There was Antony von Leeuwenhoek. He made his own microscopes, quite amazing, really. And he was the first to see and describe a live cell. Then a bit later, Robert Brown, he discovered the nucleus, that dense bit in the middle. But none of this, or very little of what we know now, would be possible without the tools, right? The invention, the constant improvement of the microscope, eventually leading to the electron microscope. That was crucial. It let us see all the tiny structural details inside.
Okay, so once we could actually see these things, how did we go from just observing them to forming a big unifying theory about life?
Right, that's where the cell theory comes in. It didn't happen overnight. Think back to 1838. You had Matthias Schleiden, a German botanist. He was looking at plants and he realizes, hey, all these plants are made of different kinds of cells. These cells form the plant tissues.
Okay, so that's the plant side.
Then, just a year later, 1839, Theodor Schwann, a zoologist this time, he's studying animal cells. He noticed they have this thin outer layer, what we now call the plasma membrane. And comparing notes with Schleiden probably, he realized the cell wall was unique to plants. So he put it together and hypothesized that, well, the bodies of both animals and plants are made of cells and their products.
So Schleiden and Schwann together formulated the first version of the cell theory. But there was a gap, wasn't there? I remember reading it didn't explain. Well, how do you get new cells?
Exactly. You got it. That was the big missing piece. How do cells reproduce? The answer came from Rudolf Virchow in 1855, a very famous statement. He explained that cells divide and new cells are formed from pre-existing cells. His Latin phrase, omnis cellulae cellula, all cells from cells that became totally central to biology.
Right. So if we put Virchow's contribution together with Schleiden and Schwann's, we get the cell theory as we understand it today. 1, all living organisms are composed of cells and products of cells. And 2, all cells arise from pre-existing cells. Simple, but profound.
Elegantly simple, yeah. But the implications are huge.
Okay, with that foundation laid, let's explore the variety, because not all cells are the same, are they? We generally talk about 2 major types, prokaryotic and eukaryotic. What's the big difference?
The main distinction, it's really about internal organization, especially the nucleus. Eukaryotic cells have a proper membrane-bound nucleus, and they also have other distinct structures wrapped in membranes. We call those organelles. Think protists, plants, animals, fungi. That's us, and most things we see.
Got it. Membrane-bound nucleus and organelles.
Yep. Prokaryotic cells, on the other hand, they lack a membrane-bound nucleus, and they lack those other membrane-bound organelles too. Your examples here are things like bacteria, blue-green algae, mycoplasma, PPLO, and generally, prokaryotes are smaller, and they multiply much, much faster than eukaryotic cells.
But despite those big differences in complexity inside, there's something universal, right? The cytoplasm.
Absolutely. In both types, prokaryotic and eukaryotic, you've got this semi-fluid matrix called cytoplasm, filling up the cell volume. And that cytoplasm, well, that's the main stage. It's where most cellular activities actually happen.
The bustling hub, as you said.
Exactly. And even with that shared basic plan, the variety is incredible. Cells differ wildly in size, shape, and what they do. You mentioned mycoplasmas, tiniest known cells, maybe 0.3 micrometers. Tiny. Then you have the other extreme, like an ostrich egg, the largest isolated single cell.
Huge contrast.
And think about nerve cells. Some are incredibly long. Their shape isn't random either. Disc-like, polygonal, columnar, irregular. The shape nearly always reflects the job they perform. Form follows function, you know?
And even within eukaryotes, there are key differences, like between plant and animal cells. You mentioned cell walls.
Right. Plant cells have cell walls, these things called plastids, like chloroplasts, and often a very large central vacuole. Animal cells typically don't have those. But animal cells usually have centrioles, which are involved in cell division, and you almost never find those in plant cells. Different toolkits for different lifestyles.
Okay, let's zoom right in now. Eukaryotic cells are famous for this internal compartmentalization, like a tiny city with specialized departments. Let's explore some of these individual organelles. What are their structures, their functions?
Yeah, let's unpack that. We can start right at the edge or just inside the wall for plants. The cell membrane, or plasma membrane, it's mainly lipids and proteins. The big players in lipids are phospholipids, arranged in that famous bilayer. Their polar heads face the watery environment outside and inside the cell, and the hydrophobic tails hide away in the middle.
Water-loving heads out, water-fearing tails in. Makes sense.
Oh, exactly. Yeah. Cholesterol is tucked in there too, helps with stability. Yeah. And the mix of protein and lipid isn't fixed. It varies. Like in human red blood cells, it's about 52% protein, 40% lipids. Different jobs need different membrane compositions.
And how do we understand this structure? The fluid mosaic model.
That's the one. Singer and Nicholson, 1972. It's the cornerstone. The key idea is that the membrane isn't rigid, it's fluid, dynamic. The lipids form this sort of sea, and the proteins are like icebergs floating in it, able to move laterally. This fluidity is vital. Think cell growth, division, forming connections between cells, secretion, even recognition.
So it's not just a passive bag, it's actively involved.
Very actively involved. And one of its biggest jobs is transport, controlling what gets in and out. There's passive transport, no energy needed, like simple diffusion for small, neutral stuff moving down a concentration gradient. Or osmosis, the movement of water.
Just flowing downhill, basically.
Pretty much. Then there's facilitated transport. This is for polar molecules that can't just slip through the lipid part. They need a helper, a carrier protein, to get across. Still passive, usually follows the gradient.
Okay, like needing a specific key for a door.
Good analogy. And finally, there's active transport. This one needs energy, usually from ATP. This lets the cell pump things against their concentration gradient, uphill. The sodium-potassium pump is a classic example, vital for nerve function.
Right, pushing things where they wouldn't naturally go.
Exactly. Now, for plants and fungi, remember they have that extra layer outside the membrane? Yeah. The cell wall. It's non-living, rigid, gives shape, protection from damage, infection. Also helps cells interact and blocks unwanted big molecules, made of stuff like cellulose pectins. And neighboring plant cells are glued by a middle lamella, but they can still communicate through tiny channels called plasmodesmata. Connects their cytoplasm.
Fascinating. Okay, moving inside the eukaryotic cell. You mentioned this endomembrane system, a team of organelles working together.
Yes, their functions are coordinated. It includes the endoplasmic reticulum, ER, the Golgi apparatus, lysosomes, and vacuoles.
Let's start with the ER.
Okay, the endoplasmic reticulum. It's this huge network of tiny tubes and sacs spreading throughout the cytoplasm. Two types, rough ER or RER. Looks rough because it's studded with ribosomes on the outside.
Ribosomes, protein factories, right?
Exactly. So the RUR is heavily involved in making proteins that are going to be secreted or embedded in membranes. It modifies them too. And interestingly, it's often continuous with the outer membrane of the nuclear envelope. Shows that connection.
And the other type, smooth ER.
Smooth ER or SCR. No, ribosomes look smooth. This is the main place for synthesizing lipids, like oils, phospholipids, and steroids. In animal cells, it makes steroid hormones. Also plays a big role in detoxifying drugs and poisons.
Okay, ER makes proteins and lipids. Then what? The Golgi apparatus.
Yep, the Golgi. Named after Camillo Golgi, who first saw it in 1898. Picture a stack of flattened, membrane-bound sacs called cisternae, kind of like pita bread stacked up. It has a receiving side, the cis face, usually near the ER, and a shipping side, the trans face.
Receiving and shipping, like a post office.
That's a great way to think about it. Yeah. It receives proteins and lipids from the ER, modifies them further, sorts them, and packages them into vesicles for delivery. It's also where glycoproteins, proteins with sugars attached, and glycolipids are made. Crucial stuff.
Okay, so ER makes, Golgi processes and ships. What about lysosomes?
Lysosomes are basically membrane sacs filled with digestive enzymes. They butt off from the Golgi. Think of them as the cell's recycling center or waste disposal system. They're packed with hydrolytic enzymes, lipases, proteases, carbohydrases that can break down all sorts of macromolecules. And importantly, these enzymes work best at an acidic pH, which is maintained inside the lysosome.
A safety feature, so they don't digest the rest of the cell.
Precisely. If a lysosome breaks, the enzymes aren't very active in the neutral pH of the cytoplasm. Clever design. They digest food particles, old organelles, even invading bacteria or viruses.
And the last part of the system, vacuoles.
Vacuoles. These are membrane-bound sacs, but their roles are more varied. They contain water, ions, pigments, waste products, nutrients. The membrane around them is called the tonoplast. In plant cells, there's often one huge central vacuole. It can take up 90% of the volume, helps maintain turgor pressure, store compounds, break down waste.
So much bigger role in plants than animals generally.
Often, yes. Though animal cells can have vacuoles, they're usually smaller and more specialized, like contractile vacuoles in protists like amoeba for pumping out excess water, or food vacuoles formed during phagocytosis.
All right, that covers the endomembrane system. What about the energy producers, mitochondria?
Ah, yes, the famous powerhouses of the cell.
All right.
Mitochondria. These are distinctive. They have two membranes, a smooth outer one and a highly folded inner membrane. Those folds are called cristae, and they dramatically increase the surface area inside.
More surface.
So the chemical reactions of aerobic respiration. That's where glucose and oxygen are used to generate ATP, the main energy currency of the cell. The inner compartment is filled with the matrix, a dense substance with enzymes, mitochondrial DNA, and ribosomes.
Wait, they have their own DNA and ribosomes?
They do. A single circular DNA molecule, similar to bacteria, and 70S ribosomes, also like bacteria. They can even divide on their own by fission. All this points to their evolutionary origin, likely engulfed bacteria long ago.
Fascinating. Okay, what about plastids? You mentioned those in plants.
Right, plastids. Found in plants and some protists, like euglena. Also, often double-membraned. We classify them based on pigments. Most famous are chloroplasts. They contain chlorophyll green and carotenoid pigments, yellow-orange. This is where photosynthesis happens, capturing light energy to make sugars.
The solar panels of the cell.
Good one. Then there are chromoplasts. They contain fat-soluble carotenoids like carotene and xanthophylls. They give the yellow, orange, and red colors to fruits, flowers, and autumn leaves.
Attracting pollinators or seed dispersers, maybe?
Likely, yes. And finally, leucoplasts. These are colorless plastids used for storage. Amyloplasts store starch, carbohydrates. Eleoplasts store oils and fats, and aleuroplasts store proteins.
So a whole family of related storage and pigment organelles. Can we look inside a chloroplast quickly?
Sure, like mitochondria, double membrane. Inside is the stroma, the fluid-filled space. Within the stroma are these flattened sacs called thylakoids. Chlorophyll is embedded in the thylakoid membranes. Thylakoids are often stacked into columns called grana, like stacks of coins, and connecting the grana are stromal lamellae. The stroma itself has enzymes, DNA, and 70S ribosomes, again hinting at that prokaryotic origin.
Okay, amazing detail. We mentioned ribosomes a few times, the protein factories. Let's focus on them for a moment.
Right, ribosomes.
Yeah.
Crucial. George Palade first observed these granular structures back in 1953. They're made of ribosomal RNA.
Yeah.
RNA. And proteins. Key thing, they are not surrounded by a membrane, found in both prokaryotes and eukaryotes.
But there's a size difference, right? 70S versus 80S.
Correct. Prokaryotic ribosomes are smaller, 70S. Eukaryotic ribosomes, found in the cytoplasm and on the RER, are larger, 80S. Remember, mitochondria and chloroplasts have 70S ones. That S is Svedberg unit measures sedimentation rate, related to size and density. And often you see several ribosomes attached to a single messenger RNA, mRNA molecule, translating it simultaneously. That structure is called a polyribosome, or polysome. Very efficient protein production line.
Makes sense. Now what holds everything together gives the cell shape, the cytoskeleton.
Exactly, the cytoskeleton. It's not just empty space in there. It's an elaborate network of protein filaments. Three main types: microtubules, hollow tubes, microfilaments, thinner, solid rods often made of actin, and intermediate filaments, fibrous proteins more stable. Together, they provide mechanical support, help maintain cell shape, anchor organelles, and are crucial for cell movement and internal transport, like railway tracks for vesicles.
And related to movement, cilia and flagella.
Yes, these are extensions from the cell surface involved in motility. Cilia are usually shorter, more numerous, and work like oars, moving the cell or moving fluid over the cell surface. Think respiratory tract lining. Flagella are longer, usually just one or a few per cell, and they have more of a whip-like motion for propulsion, like a sperm cell tail.
And inside, they have a specific structure.
They do. A core called the axonome, which has a characteristic 9+2 arrangement of microtubules, 9 pairs arranged in a circle around 2 central microtubules. These grow out from a basal body, which is structurally very similar to a centriole.
Which brings us to the centrosome and centrioles. You mentioned those are usually in animal cells.
Correct. The centrosome is the main microtubule organizing center in animal cells. It contains a pair of centrioles. These are short, cylindrical structures arranged perpendicular to each other, kind of like a T-shape. Each centriole itself has a cartwheel structure made of 9 triplets of microtubules, but no central ones, a 9+0 arrangement. They form the basal bodies of cilia and flagella, and critically, during cell division, they organize the mitotic spindle fibers that separate chromosomes.
Okay, nearly there with the organelles. What are microbodies?
Microbodies are just small, membrane-bound vesicles containing various enzymes. They're involved in different metabolic processes. Peroxisomes are a well-known example, involved in breaking down fatty acids and detoxifying substances, found in both plant and animal cells.
Wow, that is an incredibly intricate city inside each eukaryotic cell. Amazing. But now let's focus on the city hall or the command center, the nucleus.
The nucleus, yes. The control center. It was actually seen quite early on. Robert Brown described it back in 1831. Later, the stuff inside, the material that stains easily, was called chromatin by Slemming. Structurally, it's enclosed by the nuclear envelope, a double membrane. 2 parallel membranes with a space between, the paranuclear space.
A double layer of protection for the genetic material.
Exactly. It acts as a barrier. And the outer membrane, as we said, is often continuous with the ER and can have ribosomes on it. Integrated system.
But things need to get in and out, right? RNA, proteins?
Absolutely. The envelope isn't sealed. It's dotted with nuclear pores. These aren't just simple holes. They're complex structures that regulate the traffic. They act as gateways for molecules moving between the nucleus and the cytoplasm. Essential communication. And while most cells have one nucleus, it varies. Some cells have multiple nuclei. Some mature cells, like red blood cells in mammals or sieve tube cells in plants, lose their nucleus entirely.
No nucleus in red blood cells. Huh, okay. What about inside the nucleus, the nucleolus?
Right, inside the nuclear fluid, the nucleoplasm, you find one or more dense structures called nucleoli, singular, nucleolus, key point. These are not membrane-bound. They're basically dense regions of chromatin and proteins. Their main job is ribosome synthesis. Their ribosomal RNA is made and assembled with proteins to form ribosomal subunits. So cells that make a lot of protein, like muscle cells or gland cells, tend to have larger and more numerous nucleoli. Makes sense.
High demand for ribosomes. Got it. And the most famous content, chromatin and chromosomes.
The genetic material itself. In a non-dividing cell interphase, the DNA isn't just floating around loose, it's associated with proteins, forming this complex called chromatin. It looks like a diffuse, tangled mass under the microscope. The main proteins are histones, which are basic proteins that help package the DNA incredibly tightly. There are also non-histone proteins involved in gene regulation.
So chromatin is the DNA protein complex in its working state.
You can say that. But when the cell gets ready to divide, this chromatin undergoes dramatic condensation. It coils and folds up incredibly tightly to form distinct, visible structures, the chromosomes. Easier to move around without tangling. It's mind-boggling, really. A single human cell has about 2 meters of DNA packed into 46 chromosomes.
2 meters, incredible packaging and chromosome structure. The centromere.
Each condensed chromosome, after DNA replication, consists of 2 identical sister chromatids joined together. The point where they are most tightly joined is the primary constriction, called the centromere. On the surface of the centromere are these disc-like protein structures called kinetochores. These are where the spindle fibers attach during division.
And the centromere position matters.
Yes, it's used to classify chromosomes. If the centromere is right in the middle, it's metacentric, 2 equal arms. If it's slightly off-center, it's submetacentric, 1 shorter arm, 1 longer arm. If it's close to one end, it's acrocentric, 1 very short arm, 1 very long arm. And if it's right at the very end, it's telecentric, though rare in humans. Sometimes you also see secondary constrictions elsewhere on the arms, which can make a small fragment look like a satellite.
Okay, we've gone from the whole cell down to the DNA packed into chromosomes in the nucleus. Amazing detail. Let's circle back now to that idea you introduced earlier, reductionist biology. How does that approach tie all this together?
Right, reductionist biology. It's essentially this physicochemical approach to understanding life. Instead of just describing what happens, it tries to explain biological processes in terms of molecules and their interactions. Physics and chemistry applied to biology. It asks, what are these specific molecules, proteins, lipids, DNA actually doing inside the cell? What's the molecular basis for, say, muscle contraction or nerve impulses?
So digging down to the fundamental mechanisms.
Exactly. And it's incredibly powerful. It helps us understand normal physiology, but also what goes wrong at a molecular level in disease. Understanding abnormal processes during illness often comes down to understanding changes in these molecular interactions.
And you mentioned G.N. Ramachandran as a great example of someone using this approach.
Oh, absolutely. A truly outstanding figure in protein structure. He founded the Madras School of Biopolymer Conformation. He applied principles of physics and geometry to understand how protein chains could fold. His discovery of the triple helical structure of collagen, published in Nature in '54, was huge. Collagen is a major structural protein, and maybe even more famously, his analysis led to the Ramachandran plot.
The Ramachandran plot, what does that show?
It basically maps out which combinations of bond angles are physically possible, or allowed, for the amino acid backbone in a protein. It tells you which shapes a protein chain can adopt without atoms bumping into each other. Hugely influential in determining protein structures. He was inspired by people like Linus Pauling, another giant who applied chemistry to biology. It really shows how crossing disciplines leads to breakthroughs.
That's a fantastic example of reductionism in action. What an incredible journey we've taken today. We started with that big question, what makes something alive, and ended up deep inside the molecular machinery. We traced the discovery, the theory, the different types, the organelles, the nucleus. It just reinforces it at every step, doesn't it? The cell truly is the fundamental structural and functional unit of life.
It absolutely is. And perhaps a final thought for you to mull over building on all this.
As we get even better at understanding these intricate cellular details, these molecular mechanisms, how might that continue to revolutionize how we approach health and disease? Especially thinking about those abnormal processes that cause illness. Understanding the machine helps us figure out how to fix it when it breaks down, right? There's always more to uncover.